Imagine a world where we could predict a storm before the first cloud forms. We could see not just that it will rain, but precisely how much, and where. You know, in the universe of our own cells, cancer is a storm. It begins with tiny, invisible changes in our genetic code. These changes, called mutations, can be harmless static or the first rumblings of a hurricane. For a long time, we have been caught in the downpour, only able to react. A new study, published today, 2026 February 6, changes that. Scientists have created an unprecedented map, a detailed atlas of every possible mutation in a critical hotspot of a major cancer gene, giving us a weather forecast for cancer itself. This is not just another discovery. It is a fundamental shift in perspective. Think of a gene as a long sentence that tells a cell how to behave. Most of the time, we find a single typo in a patient and try to figure out what it means. It is like finding one misspelled word in a giant library and guessing its impact. This new map changes that, by charting a hotspot completely. So, how did the scientists build this extraordinary map? The method is a story of ingenuity and scale. Traditionally, studying mutations is slow and piecemeal. You find a mutation in a patient, recreate it in lab-grown cells, and observe what happens. Doing this for hundreds of mutations would take decades, so the team built something smarter. They used advanced gene editing technology to introduce every possible single-letter change inside a crucial hotspot of a cancer gene. This created a massive library of cells, each carrying a different specific mutation. Next, they measured the effect of each change. The gene sits in a pathway that tells cells when to grow and divide. Think of the pathway as a series of dominoes. In healthy cells, growth signals are tightly controlled. In cancer, mutations can push the first domino without permission, triggering uncontrolled growth. So they installed a biological meter to read signal strength from each mutation. Hundreds tested side by side under identical conditions, so effects could be directly compared. This removes guesswork that plagues one-at-a-time studies. They produced a unified map, numeric scores for each mutation, from benign to catastrophic. The results were striking. A continuous spectrum emerged, from silent to supercharged. On one end, many mutations were functionally silent, no change in growth signal. Cells behaved like normal. These are whispers, variants that can be safely ignored. On the other end, supercharging mutations, the shouts. A single change, say G to T, can boost the signal 100-fold, you know. These drive malignant growth and spread. In between lies a middle ground, mutations that double or triple the signal. They can explain slower growth or different treatment responses. A beautiful map only matters if it matches reality, so they validated against real patient data. The correlation was clear. Weak lab signals were rare in tumors, strong ones recurred across cancers. From lung to colon to brain, predictions held across tissues. This elevates the map into a clinically relevant tool. Now when a tumor report shows a hotspot variant, a doctor gets a data-backed score, not a variant of uncertain significance. Supercharging mutations point to powerful blockers. Weak mutations signal when to spare cost and side effects. Drug discovery can focus on the most potent mutations and the hard mid-levels. This map covers one hotspot in one gene. Next, build the atlas of cancer mutations across genes and hotspots.